Hershey and Chase in 1952 conducted an experiment to determine what the genetic material was in organisms. They used bacteriophages because they were simple, composed of a protein outer coat housing DNA. At the time, researchers thought it was either protein or DNA that made up the genetic material. Bacteriophages work by infecting a host. Hershey and Chase used E. coli as the host bacteria. The bacteriophages latch onto the bacterium and infect it by injecting its genetic material to create copies. This is important because they needed a way to see where the genetic material ended up. This was achieved by growing E. coli in two separate mediums, one containing radiolabeled sulfur, 35S, and the other containing radiolabeled phosphorus, 32P. The phages use these elements when replicating. Sulfur is only found in protein, and phosphorus is only found in DNA. This creates a way to determine where the protein and DNA end up at the end of the experiment. I will now demonstrate how Hershey and Chase conducted their experiment. As previously mentioned, they had two separate mediums for phosphorus and sulfur. The E. coli are represented by the oats and are allowed to grow in the medium. The phages, represented by the garlic salt, are added to the medium E. coli mixture. The mixture is then blended to shred the phage coats on the outside of the bacteria and centrifuge to separate the material. The lighter top slurry consists of the phage coats, while the heavier bottom slurry consists of the bacterial contents and genetic material. Note that both mediums were blended and centrifuged separately. When the phosphorus mixture was checked for what was radiolabeled, it showed the phosphorus P32 in the bottom slurry, indicating DNA's genetic material and is passed on to the phage progeny, while when the sulfur S35 mixture was checked, it showed the sulfur in the top slurry. This helped answer the question of their time of what is the genetic material in organisms.